Hello, I believe this is chapter 10, notes recording 74. I'm just kidding. It's notes recording, I think 5 at this point. Turns out it's actually number 6. Anyhow, alright, this first slide deals in the new concept, sister chromatid. And this is our first step to really kind of learn some language, some academic, scientific words to describe DNA in certain forms inside the cell. One form of DNA is chromosomes, and we've already kind of looked at chromosomes. Just in general, we've studied meiosis to understand segregation. We know that chromosomes are separated during meiosis to create sperm or eggs with fewer numbers of chromosomes. But we're also wanting to learn different names for specific chromosomes. Here, a sister chromatid is technically this thing right here within the yellow box. So whatever's in the yellow box would be a sister chromatid. So if you're thinking, is the other side a sister chromatid? You're correct. So when you see this X-shaped thing we're calling a chromosome, it really is the chromosome itself is actually a clone, or two identical copies we call the sister chromatid. And both of the copies are joined in the middle, right here, in a place called the centromere. That's a term I won't necessarily require you to memorize, but centromere, center of the chromosome is where two separate sister chromatids are joined. They're not forever joined, and we'll see that they actually can be pulled apart. We may have already noticed this through segregation, but part of segregation is actually being able to disconnect this sister chromatid. Keep screwing up. From this one, right? So during meiosis, in order to create segregation or to reduce the number of chromosomes in each gamete, each of these two sister chromatids have to be separated. All right. All right, to help you solve the questions on the page, the first question asks you to interpret the image, and that means to interpret means to make sense of what you see, right? To try to understand what you see. And it says A. Does A represent something called a homologous chromosome, or does A represent sister chromatids? So A would be if you took all of these things together. So every structure here, if you took them all together, would it represent a pair of homologous chromosomes? Or does this thing altogether represent sister chromatids? The next question is about B, and B is here, right here. So whatever is included within this bracket, which is this and this. So B is only this and this section here. Just looking at the blue. It says, is B representing a homologous chromosome or a sister chromatid? Hopefully, after our early description, you understand the question. You can do your best, okay? Read over the earlier minute and a half if you want to better understand the question again. Just rewind a minute and a half to feel confident about answering this. All right, to give you a little help, each of these are sister chromatids of each other. So if you're trying to answer the question from the earlier part of the video, each of these would represent sister chromatids. And if the two X-shaped structures, each of these would be considered homologous regions. Just as you see here, homologous region. So homologous chromosomes are these two, and each individual site are considered sister chromatids. This is a sister chromatid of this one only. And if you've noticed, each of these chromosomes have slightly different colors because each chromosome contains different alleles for the same gene. Here we go. It's a chance that says look at the numbers 1, 4, and 5 and predict which corresponds to each situation below. So in your notebook, in your notes, you'd write chromosomes duplicated at step 1, 4, or 5, in your notebook, you'd say chromosomes were, the duplicated chromosomes were broken apart like Kit Kats. At which step 1, 4, or 5, and then finally you would, in your notebook, identify which step, 1, 4, or 5, gets a copy of this chromosome. So, just based on what you see, what you think, which one of these steps duplicates chromosome, which one of these steps breaks chromosomes apart, like a Kit Kat, and which step ensures that each gamete gets only one number or correct number of sister chromatids. So when it says this single one, this is actually sister chromatid. It's going to be important to understand this language because in the Chapter 10 project, Day 2, it's going to ask you to use some of these words. So this would be a homologous chromosome, and these would be sister chromatids, okay? So sister chromatids are joined in the middle, but they can also be pulled apart from the center or centromere area into these individual areas. This is a normal process. All right, good luck. All right, here we have meiosis, and it asks you just a few questions using our new language of homologous chromosomes. It says, identify cell division where homologous chromosomes are separated. So which step, top, next, third, well, we could say the first, second, or third, first, second, or third, which division, the first, the second, or the third, divides and separates the homologous chromosomes. Then it says, identify the cell division separating the sister chromatids. Sister chromatids, would that be the first, second, or third separates sister chromatids? And then lastly, it says, investigate the change in chromosome number during meiosis. List 3 facts or observations describing how chromosome number is changing. However, this time when you describe how chromosome number changes, use the correct language. Discuss how homologous chromosomes and sister chromatids are involved. Try your best. Just to make sure we fully understand everything again, and to help you with that earlier question, the one we just asked, this would be a pair of homologous chromosomes. During meiosis, they're separated. And this one is supposed to go into one cell, and this other is supposed to go in a different cell that will later produce a gamete, a sperm or an egg. And then later, during meiosis, these homologous chromosomes not only are separated, but each chromosome copy is separated. In other words, the sister chromatids, which are clones of each other, here and here, will eventually be separated also into a sperm and egg. The ending result, of course, is a sperm or egg receiving fewer chromosomes than the original cell that started making them. Here's a Punnett square, and the task is, in your notes, you should be writing this setup. So you should write draw the 2 eggs and the 2 sperm on top, 2 sperm from father, but inside show the chromosome, which would be a sister chromatid, show this sister chromatid and the allele that it contains, and this sister chromatid and the allele it contains, and then set up the egg, sister chromatid with the allele it contains, and the sister chromatid with the allele it contains. And your job is, in the inside of the square, to correctly fill it in, showing a larger cell that has both sister chromatids for each baby. So you have 4 boxes to fill in, and you should show how when the sperm and egg fuse, you get the sister chromatid from the egg and the sister chromatid from the father combining together to provide the 2 chromosomes for whichever chromosome number it would be. It'll look something like that. It's a brief reference. Hi, we have this time an official definition for homologous chromosomes. It says 2 chromosomes containing the same gene or a trait. Now, If they contain the same genes for a trait, it doesn't mean that they have the same versions or allele. It just means they have the same gene. So it must be dominant or recessive allele for the same gene, but they don't have to match. An example is in this picture. You see this, there's a pair of homologous chromosomes here. They are homologous because each sister chromatid contains a B gene. It doesn't have to be, they don't both have to be dominant, they both don't have to be recessive. As long as each chromosome contains a B gene, the two chromosomes are homologous, and they would match up during meiosis to produce sperm or egg. So the question is, in this picture, are each of these homologous to one another? In other words, do they contain the same genes? If they do, you would call them homologous. You decide, as your hypothesis, in your notebook, my hypothesis is, and then lastly, you support it with evidence. What made you think it is or isn't homologous? This question is right out of the textbook, so if you turn to chapter 10, you'll find this if you want to see it in a little more zoomed in or easier to read. But each of these 4 questions I expect you to be able to answer, and it should be in your notebook. Here it says, could chromosome 2, which is this guy, be homologous with chromosome 1? So you compare the two chromosomes and you have to decide, are they homologous? Well, I'm going to help you with number 1, and you'll do 2 through 4 on your own. Here, they both contain the F gene, both contain the G gene, both contain the H, both contain the J. Although they have different versions of each of these genes, they all contain the same 4 genes. Because they contain the same 4 genes, even if they're different versions or alleles, they are homologous. So chromosomes 2 and 1 are homologous because they each contain the F, the G, the H, and the J gene. All right, complete 2 through 4 on your own by comparing 1 to chromosome 3, 4, or 5. All right, this is bringing us back to Chapter 10 project. And your Chapter 10 project, you need to complete the page 2 of your first doctor's worksheet, where you're going to properly fill this section out and this section out using language and knowledge of what we've just learned, as well as going back to the Learn Genetics website to cover some of the causes of your genetic disorder that you have diagnosed. So when you're trying to complete page 2, again, you need to go back to genetic disorders, go to chromosome abnormalities, and you pick whichever disorder that you have diagnosed your patient with. I'm going to continue giving my example, which was Down syndrome. And you click on Down syndrome, and it will provide you, and the same for the other syndromes. It'll show you with the other syndromes what it led to. And it says having an extra copy of chromosome 21. And the person had overall 3 copies of that chromosome. Right, so genes on chromosome 21, if you get too many of the chromosome 21, which is highlighted in the circle, you get Down syndrome. So how would that look like? So we know a sperm or an egg. We have to decide who we're blaming, mom or dad. But how would we show the child receiving 3? Well, I have an example, and I'll show you how I did for Down syndrome, and then you'll need to repeat that on your own for your own disorder and whatever caused your own, not disorder, but syndrome. All right. So again, my Down syndrome example is only an example. You'll need to figure out and adapt it for your own syndrome and what you've diagnosed for your patient. So this is a part of the page 2 that you need to do for your research, and it should look familiar. On this slide, I only included a portion of the task because that's what I'm most concerned with. I show you right here, you need to identify what chromosome is involved in your disorder. Is it a sex chromosome or is it a non-sex chromosome? In this example for Down syndrome, it's chromosome 21. I showed you what a homologous pair looks like, and then I show the homologous pair being divided, and then it's separating into sister chromatids, but improperly. Well, you got one, in this example, an egg. Notice they don't have tails at the bottom, so it has to be an egg, so mom's responsible. Mom's egg improperly divided sister chromatids, and the egg received 2. Meanwhile, one received none because they failed to divide properly. And then you had 2 eggs that received the normal amount. And then when we take mom's 2 eggs here, or one, depending on you, and you set it up properly, assuming dad is normal with one